Still had a lab there.
I appreciate it.
Oh, thank you.
You like my kids?
It's a number one favorite.
What?
It's my number one favorite.
Oh, okay.
Mhm.
Actually,
Well,
just realized I didn't turn on the screen. Almost there. 3 more days of lecture, 1 lab and 1 final. We're kind of extra final. And hopefully it hasn't been too bad. I know it's not, I didn't get near enough accomplished, but hopefully most of what I've discussed has made sense. I didn't got that set up either. Goodness, I'm following that one along. Okay, folks, unfortunately, you're stuck listening to my voice, but it's not part of the. I'm going to try to minimize excess noise. So we really started the beginning of the semester off doing organic reactions. The organic reactions were smaller versions of the chemical reactions that are associated with the biological molecules that we work with. We've seen this already when we dealt with the protein formation through the peptide bonds. We've seen this already as we talked about hemiacetal formation in the sugars. So you've seen how those organic reactions were directly applicable to some of the chemistry associated with biological molecules. And we've slowly been the last few chapters has slowly been building up this concept of the biological molecules by talking about protein formation, and then enzymes and how enzymes operate, and then into the carbohydrates and some of the reactions carbohydrates undergo. Now we're going to take and we're going to combine many of these concepts, especially the concepts of enzymes and the concepts of the carbohydrates, and we're really going to start getting into the generation of biochemical energy. Energy is required for all of life's functions. It is required to maintain the body temperature that our bodies possess. It's required for us to walk, talk, you name it. Energy is going to be required. Our body, inside our body, requires energy as well in order to perform the chemical processes that the enzymes initiate in the active site. It is going to be required in order to move molecules across cell walls. So it's required for everything. And so we need to talk about the formation of this energy in the biochemical systems. Slide. All of the energy that we obtain has to come from the food that we eat, but we cannot, the food that we eat is really high in energy, and we cannot absorb all of that energy in one fell swoop. We have to, when we eat, we'll use a certain amount of that food to provide the energy to heat our bodies, allow us to do some of the functions. But the majority of that food is going to get stored so that it can be broken down at the appropriate time that our body needs that energy. That is why a high fat or high carbohydrate diet is bad, because again, our body uses some of that energy when we digest the food, but then it has to store it. And if we have a high carbohydrate, high fat diet, it's going to store that energy, and we may or may not use that energy. So now our body breaks down this food through combustion. And like I said, it generates a lot of energy when it initially combusts. But we store some of it in accessible form so that we can use the energy when it's needed instead of trying to use it all at once. Any questions so far? The release of energy from storage must be finely controlled so that it's available when and where it is needed within the body. And we must release just the right amount so that we don't cause an excess of energy to be released at any one time. Because if we have an excess of energy that is released at any one time, our temperature would have to be the thing that has to skyrocket. Because again, those enzymes, they can only use a certain amount of energy. They only have a certain turnover rate. They're not going to speed up if you add heat to them, if they're already at their maximum turnover rate. So that extra energy would wind up going to heat our body, and we don't want it to go past a certain level or we kill cells. So there is this balance that we must maintain. We need to release the energy from storage in small quantities so that our body can keep functioning, but not over-release energy. Otherwise, our body temperature would increase, and that would be bad. Now, the energy that is released besides body temperature, like I said, it goes to help the enzymes. We talked about how enzymes perform at a specific temperature. Most of the enzymes perform best at a temperature very close to body temperature. We're going to talk about the fact that some reactions in our body are not favorable processes, and therefore the energy that is released helps to drive those unfavorable processes. So you've probably seen a similar diagram in many textbooks because you're all into something to deal with biology and energy. So I'm sure you've seen this type of diagram or something similar in many textbooks. So plants undergo photosynthesis. Photosynthesis itself is not a favorable process. Because photosynthesis is not a favorable process, it requires the sun's energy in order to cause it to occur. Once the photosynthesis of the plants occur, animals eat those plants, digest those plants, and that is converted into chemical energy. And this, no matter what type of animal, would be a favorable process, because in this process, you're going to take the plants, you're going to add oxygen to it, and you're going to digest it to form CO2 and water, which is a favorable process. Now, with respect to the animals, they again have to, the energy that we give off as we digest food is going to go to a variety of tasks. Some of those tasks are to maintain body temperature. Some of it is mechanical work, walking, talking, jogging, you name it. Anything your body does that's a mechanical process. Some of it is going to be used in our body to help do some chemical synthesis or transport things across the cell membrane. And yes, some of it is going to get lost to the surroundings because each of our bodies give off heat. That heat is lost to the surroundings. Our body must make more heat in order to maintain that temperature. So back in chapter 7 of your General Chemistry for Health Sciences course, you talked about chemical energy and you talked about the fact that chemical energy is in two forms. It's either in the form of heat, which we describe as the enthalpy of the reaction, or it is in the form of spontaneity, which we describe as entropy of the reaction. A favorable process would be one where Gibbs free energy is a negative value. An unfavorable process is where Gibbs free energy is a positive value. In our body, folks, if there is a process that is unfavorable, we have to couple that process with a favorable process in order to drive that reaction. Now, up here, I make sure that I emphasize not to confuse exergonic and exothermic. Exothermic is enthalpy. It is heat energy. Exergonic is the overall energy. It's Gibbs free energy. So if you have a negative delta G, it's an exergonic process. Whereas if it's negative delta H, it's an exothermic process. So don't confuse the two, because you can have an exothermic process and still have an endergonic reaction. Exothermic processes do not automatically mean that you're going to have an exergonic reaction. They are separate. They are two different things. Exothermic describes heat energy or the enthalpy of the reaction, whereas exergonic focuses on the Gibbs free energy. And it takes into account both the enthalpy of the reaction, the heat of the reaction, as well as the degree of disorder caused by the reaction. We're going to see how long ago today. Any questions so far? Oh, I need to send around the sign-in sheet, don't I? I forgot on Tuesday. First Tuesday, I thought was Thursday, so you never know. I had an interesting Tuesday. Okay. An endergonic reaction, meaning you have a positive delta G, is going to be an unfavorable reaction in which energy has to be absorbed by the reaction in order to make the reaction go. In other words, it's not going to go on its own. It does not like to go from reactants to products if it's endergonic. It has to be forced to do that process. Again, endothermic does not mean endergonic. Endothermic is a way to describe the enthalpy of the reaction. A positive delta H is endothermic. Endergonic means the delta G is positive. Just because the enthalpy is positive does not mean that the delta G is positive, because delta G it is associated with two things. It's associated with the enthalpy, but it's also associated with that degree of disorder. So one does not necessarily equate to the other. So when we're talking about inorganic or exergonic, what does it really mean with respect to our reactions and their free energy? So on the x-axis of this graph, it's talking about the reaction progress as you go from reactants to products. If you're and then on the y-axis, it's looking at free energy, which is Gibbs free energy. When you're talking about an exergonic reaction, it means that the reactants are higher in energy than are the products. And so you get a negative Gibbs free energy. An inorganic reaction means that the products are higher in energy than are the reactants. And so you get a positive delta G. So, folks, this may seem like, why am I going over this? But again, we have to understand this because we need to know, because in our bodies, some reactions are exergonic. That means those reactions will occur without help. But some reactions are inorganic. And if it's an inorganic reaction, that means we're going to have to couple it with an exergonic reaction that is higher in energy in order to drive the reaction. So, photosynthesis, we talked about plants. Plants take carbon dioxide and water, they synthesize them into glucose and oxygen. This process of photosynthesis is an endergonic process. It is not going to occur without help. The sun is the driving force. It provides the sun's energy is coupled with this reaction. And the sun provides the energy to overcompensate for that 686 kilocalories per mole to make that process occur. The reverse of this, however, taking glucose and reacting it with oxygen to form water and carbon dioxide, that's an endergonic process. That's something our bodies can do. And so you'll notice that the same reaction going one way is going to be a positive value and going the other way is going to be a negative value. The values are the same as long as the reaction is the same. It's just which way is favorable compared to which way is unfavorable. Again, I imagine you saw this back in chapter 7 in Gen Chem for Health Sciences. So hopefully this discussion is not entirely new. And I will post these slides today because I don't think I've posted these yet. I'll get chapter 21 posted today, along with your homework from Tuesday, homework from today, and quiz that's due Tuesday. I think that will constitute everything besides your last lab. And I'll also have all your lab grades ready to post. So I think I'm getting down to the last little bit. Okay. because we're starting to get into biochemical energy, we have to look at where the biochemical energy is produced, and that is produced in our cells. There are two main categories of cells, the prokaryotic and the eukaryotic cells. The prokaryotic cells are found in single cell organisms that include like algae and blue-green algae. Whereas the eukaryotic cells are found in some single cell organisms, but they're also found in all plants and animals. So what we're going to be focused on is the eukaryotic cell. We're not really going to be focused on the prokaryotic cell. So here's a diagram of the cell. First of all, you have your cell membrane. Your cell membrane is going to be a bilipid layer. And in that cell membrane, there are going to be some channels that allow flow of certain ions in and out of the cell, certain molecules in and out of the cell. Once you get into the cell, the majority of the cell is comprised of cytosol, which is essentially the liquid that everything resides in. You do have the nucleus along with the endoplasmic reticulum that determine the cell. When we were doing the DNA extraction the other day, what we were doing is we were breaking down the cell wall, we were breaking down the nucleus, we were with the lysine agent, which is in part detergent, in part probably there was some sand to it or some gritty material. We were breaking down those cell walls so that we could release the DNA from the plant. But the thing that we're going to focus most of our attention on in the cell is going to be the mitochondrion, because that is what that is where the energy is formed, is in the mitochondrion. Or mitochondria, they're called the power planes. They actually produce about 90% of the body's energy carrying molecule, ATP. You do have, so let's get into the mitochondrion. You have an outer membrane, you have an inner membrane, and you have intermembrane space. Within the intermembrane space, that is where your ADP, ATP conversion occurs. You have your matrix. And then you have, so that is where you, in the matrix is where the citric acid cycle occurs. And then the ADP plus phosphate goes to enzymes and coenzymes, and that goes to the cristae. And within that area also, you're going to find that that is where we do the proton transfer or transport chain. So again, we're focused mainly on the mitochondria, and we're focused in the matrix. That's where the majority of our energy is going to come from. Now, as far as the mitochondria, the majority or the greatest number of mitochondria is found in the eye, the brain, the heart, and muscle cells. In other words, the cells that are used the most, that's where you're going to find the most mitochondria too, because those are going to be the locations where we need the greatest amount of energy is in those locations. And so it is basically the number of mitochondria is directly related to how much energy we need in a particular location of our body. Okay. So again, in the mitochondria, that is where the majority of the energy is produced. So let's get into an overview of metabolism and energy production. There are a number of metabolic pathways that occur within our body that allow us to produce energy. You can have a linear sequence. The glycolysis chain is considered to be a linear sequence where we start with we take glucose and we phosphorylate glucose to make glucose 6-phosphate. The glucose 6-phosphate then enters into about a 10-step chain that then goes into the citric acid cycle. The citric acid cycle is a cyclic sequence. And in the cyclic sequence, there's actually more steps in the citric acid cycle than in this cyclic sequence. But the cyclic sequence, again, is where we can produce some of our energy. Now, one we will not get into is the spiral sequence. The spiral sequence is used to take lipids or your fats and convert those fats into the acetyl-CoA that can enter into the citric acid cycle. But we will talk about these two. You will notice that in each of these steps, an enzyme comes into play, and a lot of times a cofactor will come into play. So in other words, the formation of energy in our body is not simply going to be a single process. A lot of times it is going to be a multitude of processes. It just depends upon how our body needs to break down the food that we eat in order to get it into the citric acid cycle, in order to get it into the electron transport chain. Those are the processes that we're going to use. So for sugars or carbohydrates, it's going to be a linear sequence followed by a cyclic sequence that goes into the electron transport chain. Whereas if we were working with lipids, it would be a spiral sequence that would go into the cyclic sequence before it went into the electron transport chain. Whoops, too much. So when we talk about metabolic reaction pathways, metabolic reaction pathways can be divided into catabolism and anabolism. Catabolism occurs when you take a larger molecule and you break it down into smaller molecules. Catabolism is what is occurring when our body uses the food that we eat or the stored food and puts it into the sequences that convert it into the citric acid cycle that go into the electron transport chain. That is going to be a process of catabolism. However, as I've already stated, all of the food that you eat cannot be converted directly into energy. Some of that food has to be stored because our bodies can't handle the amount of energy that you can get from a single morsel of food in one fell swoop. So the other process is anabolism. Anabolism will take the smaller molecules. You have to add energy to the smaller molecules in order to get them into bigger molecules. So in other words, we have to take our excess glucose that we put into our body, we have to add energy to it in order to store it as starch or glycogen that can be later used by our body in metabolism. So our body does have stages that it follows in order to take food that we digest and convert it into energy. The first stage is digestion. So when you digest food, some of it's going to convert to lipids, carbohydrates, and proteins. Depending upon which form of material or compound you have, whether you have lipids, carbohydrates, or proteins, determines which pathway it takes to get into the citric acid cycle. So the second stage of this process is the formation of acetyl-CoA. And if you start out with the lipids, it's going to go through the spiral fatty acid cycle in order to convert those lipids into acetyl-CoA in order to create energy. If you talk about carbohydrates, carbohydrates are going to go through the glycolysis network, which is like a 10-step process in order to convert it ultimately into acetyl-CoA, which can enter into the citric acid cycle. The only substance that does not have to go strictly into the form of acetyl-CoA in order to enter the citric acid cycle is the breakdown of proteins. Proteins can be broken down into pyruvate, they can be broken down into the acetyl-CoA, or they can follow a pathway that will actually enter into the citric acid cycle at another location beside it as acetyl-CoA. Once you go through the citric acid cycle, that is stage 3. That is where a carbon dioxide is formed. You're going to make some ATP there. But then you're also going to make some reduced coenzymes. So NADH and the like. Those reduced coenzymes are then going to enter into the 4th stage of production, which is the electron transport chain. And it's in the electron transport chain that we absorb oxygen and we make water and additional ATPs. Folks, making those ATPs along the way is extremely important because, as we'll discuss, the formation of ATP is really, I shouldn't say the formation, the use of ATP is really the driving factor behind a lot of reactions that our body does. It's there in the happy middle ground of energy formation. This diagram, by the way, is found on page 666 of your textbook. (666). Most of what I've just asked you is going to be in the form of multiple choice, or most of what I've just discussed is going to be in the form of multiple choice, including that idea of what is a spiral sequence, what is a linear sequence, what is a cyclic sequence. Those things can be asked in the form in the multiple choice section. So just understand that in a linear sequence, we go A to B to C to D. We don't ever back up. And in a cyclic sequence, you start with one material and ultimately you remake that material. And it can go through a cycle. Trying to let those that are writing catch up to me. Because this slide has a lot of information on it. Any questions about this slide? Okay, so I keep talking about ATP. ATP stands for adenosine triphosphate. Triphosphate, meaning it has 3 phosphate groups attached to adenosine. When you break a bond, it gives off energy. That's always true. You break a bond, it gives off energy. So, when ATP is used, this last phosphate group gets broken. And therefore it produces energy. ATP, so you have energy from food that comes in. It allows to break those bonds. And as you break the bond, that some of that energy goes off for work or to heat the body. And you also will make a phosphate group. Remember transferase, which transfers a phosphate kinase, transferase kinases, which transfer a phosphate group. They use ATP for that transfer of a phosphate group. The transfer of the phosphate group out leaves adenosine diphosphate. Adenosine diphosphate, then you can bring in energy and another phosphate to remake that bond. So again, breaking ATP ultimately to ADP provides energy, but in order to remake ATP, we're going to have to add some energy in to make that ATP. What we're going to find is so ATP can be made in several locations throughout the glycolysis chain, the citric acid cycle, but the majority of ATP is going to be made in the electron transport chain. That's where we're going to get the majority of our ATP from. And this ATP can then be transferred to wherever it needs to be transferred in the body in order to provide energy for another reaction to occur. Because again, one of the ways we can make something happen is to take a reaction that gives off energy, such as this breaking down of ATP to form ADP. We give off energy. That energy that's given off can drive another reaction. that would not necessarily be a favorable process. So this formation of ATP is extremely important because it tends to be the driving force behind a lot of reactions. Why is it the driving force? Why is it the principal energy carrier? If you look at Gibbs free energy, you'll notice that ATP is in the happy spot. It has ATP to ADP has is an exergonic process that forms 7.3 kilojoules or kilojoules per mole of energy. Which, if you look at glucose 6-phosphate, the formation of glucose 6-phosphate, it's which is the first intermediate glycolysis, is a negative 3.3. But that's glucose 6-phosphate that's doing something. If glucose 6-phosphate does something, it gives off negative 3.3. But to make glucose 6-phosphate in the first place, it's not a favorable process. So we couple the glucose 6-phosphate formation with a favorable process such as ATP in order to drive the reaction. Now, ATP to ADP, if we want to do the reverse of this, we have to couple it with something that's stronger. So let me just kind of draw this up here. I can figure out which of these is the best of these markers. Let's say we're going ADP to ATP. If you do ADP to ATP, it's the reverse of what they say up here. So since it's the reverse of what they say up here, instead of having a negative, it's going to be a positive. So converting ADP to ATP is not going to happen. It is an endergonic process. All by its little lonesome, it is not going to happen. So we're going to couple it with something, such as this 1,3-bisphosphoglycerate. And when we couple that, if we look at the 1,3-bisphosphoglycerate, it has a negative 11.8 kcals per mole. This is an exergonic process. Coupling these together, you have a negative 4.5 kcals per mole that can drive, that is the result. So the coupled reaction is exergonic, and therefore it would drive the reaction to make it occur. If we take glucose and make glucose 6-phosphate, that is going to be an endergonic process that is going to be a plus 3.3 kcal per mole. We can couple that with the ATP going to form ADP, which gives off 7.3 kcals per mole. And now we're looking at a negative 4.0 kcals per mole, which makes it favorable. So coupling reactions is a common way to drive a reaction process. I can't move it. So this basically is stating what I've already stated. In our citric acid cycle and our electron transport chain, and I think even in the glycolysis network, we make ATP from ADP. Formation of ATP from ADP is not a favorable process. So one of the metabolic pathways, or one of the metabolic methods that we use to counterbalance this is that we'll couple a reaction to make the reaction favorable. So again, this is just putting into words what I've put up here in examples. And this is giving you an example that uses phosphoenol pyruvate to make pyruvate, which is actually an important reaction that can derive the formation of ATP. How would you see this on an exam? I will give you a couple of reactions, and I will ask you how you can combine the reactions to drive as a strategy to drive the process. Basically, you got to make sure it comes out negative, or I'll ask, would these two combine together in the way that they're written, would they be a favorable process? Something like that. And basically, you're looking to determine whether it's exergonic or endergonic when you couple the reactions. Now, when we do this exergonic reaction, again, this synergy is either going to be used as heat, or it's going to be used for chemical potential energy. In other words, it's going to be used to drive other reactions that may be endergonic. So the chemical energy and products of the chemical reaction, meaning in this particular case, the glucose 6-phosphate and the ADP, the chemical energy that's stored in those particular substances, along with the heat that they release, the negative 4.0 kilojoules, is ultimately, those two things are equal to the energy released in the exergonic reaction. Okay, another strategy used to deal with chemical reactions to make them favorable process is going to be the oxidized and reduced coenzymes. So adding a cofactor in the form of a coenzyme and having it undergo oxidation and reduction. This is a reminder of well, first of all, the net result of catabolism is the oxidation of food to release energy. Whereas the anabolism would be the reduction of food to make stored energy. Many metabolic reactions involve oxidation reduction reactions. As we go through the citric acid cycle, we're going to encounter NAD plus going to form NADH. This is the oxidized form. And this is the reduced form. Or we may encounter NADP plus and NADPH. Again, NADP plus is the oxidized form, whereas the NADPH is the reduced form. Oxidation involves the loss of electron. This is as a reminder. And these folks, we're going to see the reaction driven this direction in the citric acid cycle. And in the glycolysis chain. But we're going to see this direction in the electron transport chain. Now, just as a reminder of oxidation and reduction, since we're using these terms, oxidized means a loss of electrons, a loss of hydrogen, or a gain of oxygen. Whereas reduction means a gain of electrons, a gain of hydrogen, or a loss of oxygen. So since these are gaining hydrogen, they are being reduced. These are being reduced to form the reduced form. So this is reduction, whereas the opposite direction would be oxidation. Any questions? So again, oxidation reduction is going to be another major strategy. So we have two major strategies that we use in our metabolism. ATP, ADP, and oxidation reduction of coenzymes. This ultimately is saying some of the stuff that I was, and I forgot FAD. FAD is another one that's common. A steady, basically, when we're talking about the reduced coenzyme, we have a steady supply of oxidizing and reducing agents that must be present so that the enzymes can continuously cycle through this oxidized or reduced form. The ones that are most common are going to be these NAD plus, NADP plus, FAD. Those are most common. And when you reduce them, a lot of times they are also have an H plus because the signs have to be, the charges have to be the same on both sides. So you wind up with a NADPH plus, or NADPH H plus, and NADH H plus, or FADH2. Okay. So what have we talked about today? Really can be boiled down into a few things. Number one is energy. All reactions involve energy. Our body requires energy, but it requires it to be done in a very methodical and very specific quantity at a time. You can't have too much energy given off at any one time, because if you do, your temperature will increase above what it should be. We talked about the fact that this energy that we're talking about is really described by delta G, Gibbs free energy. How much free energy? It's not the enthalpy, it's not the entropy, it is the free energy that we want to focus on. And biochemical reactions are driven by this free energy. And we can state whether it's endergonic or whether it's exergonic. It'll be endergonic if it's a positive delta G, and it'll be exergonic if it's a negative delta G. We then briefly looked at the cell structure, but again, the most important part of the cell for our purposes is the mitochondria. That's where the citric acid cycle occurs. And that's where the electron transport chain is. And so that is where the majority of our energy is formed. And that's why it's considered to be the energy powerhouse of the cell. We then looked at a brief overview of the metabolic pathways that can be taken in order to provide energy. You can have a linear metabolic pathway, which is what the glycolysis chain is. You can have a cyclic metabolic pathway, which is what the citric acid cycle is. And you can have a spiral metabolic pathway, which we're not actually going to use, but that's how we would use, what we would use if we were talking about you taking lipids and converting them into energy. There are 4 stages associated with our body's conversion of food to energy. The first stage is digestion, basically going from our mouth, digesting the food to form either carbohydrates, lipids, or proteins. And depending upon which type of substance it forms will determine what chain it follows in order to get to the next stage. The next stage is its chain to convert it from the particular substance into acetyl-CoA most of the time, acetyl-S-CoA. Because the acetyl-S-CoA then can enter into the citric acid cycle, which is stage 3. During the citric acid cycle, you're definitely going to make some reduced coenzymes that will then enter into and ADP. That's going to enter into the electron transport chain, which is stage 4. And in the electron transport chain, the reduced coenzymes are going to be oxidized back to their original form so that they can go back and do what they're supposed to do in the citric acid cycle. And you're also going to make additional ATP. We also talked about strategies for metabolism. One of those strategies is coupling. Well, it's ATP. ATP is used because it is the nice middle happy ground with respect to energy that it forms from reaction. But we talked about other things such as coupling reactions to drive a reaction forward that wouldn't otherwise want to go forward. And then we talked about the second metabolic pathway, which involves our reduced coenzymes. And these reduced coenzymes are basically constantly cycling from reduced to oxidized. And we have energy that is going to help with those processes that we're going to encounter a lot of times we take the oxidized form of the coenzyme, and that is the reaction that we couple with another reaction in order to drive that reaction forward. As we couple the the oxidized coenzyme, it winds up producing a reduced coenzyme. The reduced coenzyme then goes into the electron transport chain. In the electron transport chain, our body has to add energy for the majority of that process. And that reduced coenzyme is then reoxidized to form the oxidized form of the coenzyme, which can go back up and do its functions. What we're going to talk about heavily on Tuesday of next week is the citric acid cycle, and we are going to break the citric acid cycle down into each individual step that it undergoes, which means we're going to talk about the enzymes involved. And then we'll talk about the electron transport chain probably on Thursday. So I expect literally the citric acid cycle to take us all of Tuesday. And then Thursday, I'll talk about the electron transport chain and hopefully a little bit on glycolysis. So let me just, got a couple of minutes. I want to just clarify some things that I have mentioned. Number 1, I have your last 2 labs graded. So I have the fat content lab graded. I will be posting those grades today. I also have the strawberry DNA lab graded. For those that have turned it in, I know it's not due until Monday, so if you haven't turned it in, it's fine. But I have graded the ones that have been turned in, and I will post that grade today as well. I will be posting 2 homeworks. One about the material that we went over on Tuesday, which had to deal with carbohydrates. And then one over these metabolic pathways, these strategies, and that type of thing for today's. I will also be posting a quiz today, and you'll have until Tuesday to complete the quiz. You'll have until Monday to complete the homeworks, like I've been doing. If I'm not mistaken, your 2 lab grades, the 2 homeworks, and the quiz grade is the last of the grades other than your lab grade on Monday. Now, Monday's lab is actually a completion lab. If you're there and you do the lab, and you do the post lab and all of that, I will look over it. And I mean, you can turn it in on Thursday because I believe I said you could, but you can also turn it in on Monday. And if you turn it in on Monday, I'm just looking for completion and that you did put forth the effort. So anybody that completes it and put forth the effort is going to get their full 25. And then that will leave just the exam. I will next week have a study guide for exam 4 that I'll bring to class so that you have an idea of what type of questions you can consider for exam 4 for those that are taking it. What I will ask is if you decide you're not taking exam 4 after all the grades are in, on Thursday of next week, when I pass around the sign-in sheet, I'll reiterate this, but if you're not taking the final, I'll probably have you star your name so that I don't overprint the exam just to make it where I'm not wasting paper. Anyhow, any questions over what's left?
Like you drop, like, I know you said you're dropping a couple things, so before we go in and take the final, should we like calculate it on our own, drop our loads?
If you want, I've got to do some, I haven't mentioned this yet, so thank you for asking. I've got to check, but I think I have it set up where it'll show you what your grade will be if you drop the final. It's still going to change until I record the last of your labs. It's still going to change, in other words, through at least Tuesday of next week. But I do have a column that shows you how many, so I'll have 2 columns that will post probably Tuesday of next week. One will show you what your grade would be if you didn't drop the lab or didn't drop the final. I'm sorry. One will show you what your grade will be if you drop the final. The other one will show you how many points you have going into the final. So you can estimate how many points you need in order to get an A, B, and C.
Right, so, but drop in like, I don't know, our lowest lab or something, do we calculate?
I've already done that in these columns.
Okay.
So it'll show you everything. It literally will be down to, this would be your grade if you don't come to the final. This is how many points you have if you do come to the final, so you can estimate what you need on the final to get a B or C or an A. So it'll be there, but I have to wait until Tuesday next week because I won't have all your grades really until then. Any other questions? Okay, I will be in my office until 11:45 this morning. And I will be back in my office from 3 to 4 this afternoon. I think today was the last day to bring food. if I recall correctly, for the canned food drive. And I'd rather you bring it up there so that I don't have to try to carry it all. And I need you to put your name on the food that you're bringing. I'll shut down and I'll set right on up.
I have to purchase now an email about next week.
Yeah.
It's very stressful, so, but this class is a lot easier, especially since we have the few reports, so I'll be able to watch those and then I'll be able to get the study guide off whenever you post the key.
Yeah, I can do that. I can post the study guide too, so that you can try it yourself and then look at the key.
Okay, yeah, because I'll be able to go to the lab on Monday, but we leave at 3 o'clock in the morning on Tuesday, so, and then get back late Saturday.
That is bad timing.
It really is, because last year they had it the week before study week. And so we at least had time to come back and have that whole week to study, but not this year.